The topic of my today's presentation or lecture is assessment of differentially expressed genetic elements responsible for salt tolerance in canola, that is Brassica napus, through RNA-Seq analysis. Before going into detail, I want to introduce to the problem of salinity. It is a worldwide problem and a serious obstacle for agricultural crops. And 26% cultivated land is salt affected in Pakistan. Various plant species can survive certain levels of salinity in the rooting medium, but the rest cannot. The plants that can tolerate salinity are known as halophytes, but the plants that cannot tolerate are known as lycophytes. Even among the glycophytic species that cannot tolerate higher levels of salinity in the rooting medium of the plants, even they have some cultivars or varieties among them that have some potential to grow on salt-affected lands. If the plants are grown on salt-affected areas or salinity stress is subjected to the growing plants, what happens? What are the different consequences? Main effects that appear on the plant growth are that salinity stress causes decrease in water potential. It also causes iron toxicity. It triggers mineral nutrition imbalance and changes the level of activities of different hormones. And it also results in the accumulation of different reactive oxygen species or ROs. And they in turn deteriorate certain membranous structures in the plant cell. And when the thylakoid is affected, they cause reduction in the process of photosynthesis. If we want to exploit such salt-affected land, we will need to grow different crops that have some salt tolerance. Certain species like barley, wheat, and brassicas, they have some salt-tolerant cultivars among them, and selecting relatively more salt-tolerant genotypes is a good strategy to utilize such salt-affected areas. And if you want to utilize that area, you have to screen different available varieties or cultivars so that you can select over some tolerant varieties or cultivars that can appear more appropriate for cultivation. And for the selection purpose, we can go for agronomic attributes, which most of the agricultural practitioners do, or we can go for some physiological attributes. We have discussed that salt stress has some consequences on the growth of plants because basically, if you know the physiology behind, plants uptake water from the rooting medium, and that has to go from a higher concentration of salts of water to the lower concentration of water, or lower concentration of salts to higher concentration of salt. Because the plant sap has higher concentration of salts, water is uptaken by the roots. If the salinity is prevailing and there is a higher salt content or concentration on the outer environment in the rooting medium, the plants will not be able to uptake the water. First of all, that will tend to decrease the water potential inside the plant, and they will be they will undergo wilting process, and they can revive certain levels of wilting and until they reach permanent wilting point from where they cannot revive back. And this decrease in water potential is very crucial for all plants. If they are somehow able to take up water, they have to accept unwanted salts along with that. And that uptake of unwanted or toxic soils is called as iron toxicity that will be generated within the cells of the plant because they were not required, but osmotically due to regulate the uptake of water, they had to take up those salts. Then if these salts are taken up, they trigger the nutritional imbalance. Some of the required minerals are not being uptaken. These all changes trigger certain other changes like activities of hormones are changed, Then they trigger production of certain reactive oxygen species, and reactive oxygen species cause oxidative stress, and they damage different membranous structures in the plant cells. And even the thylakoids are the membranous organizations, and they are disrupted and hence result in the reduction of overall photosynthesis. And these photosynthetic plants or organisms are the main accumulators of carbon dioxide in the form of carbohydrates. When the process of photosynthesis stops, initially the growth of the plants is stopped. Then finally, overall productivity of the crop and the globe is, you can say, that is harmed. If we want to change this setup and we want to utilize the higher uptake concentration of the salts in the rooting medium, you have to take up or plant those crops or cultivars of lycophytes that are able to not only survive in the saline medium, or they are able to regulate their biochemistry, physiology, and genetics in such a way that they are able to tolerate these all negative effects of salinity stress. Next slide, please. This is, I think I have already discussed that different selection criteria are settled down by different physiologists and agronomists, that they consider certain traits to screen out different available varieties that will tell you the performance of certain species under saline conditions. For example, barley, wheat, and brassicas, they have certain genotypes which can be utilized. Other approaches that can be utilized include that we can use certain genetic or non-genic markers that can be utilized by different plant breeders and different scientists that are involved in developing certain tolerant varieties. They can incorporate directly those genetic information into the genomes of the same species or other related relatives of those species to make more tolerant crops. That is the objective that our research will lead to. Next. Our selection of the crop was based on its importance. It's a potential oilseed crop, canola, that is Brassica napus, and it is moderately salt tolerant, and it is third potential oilseed crop following soybean and palm oil. It has a good quality seed oil, and it has low saturated fatty acid contents among major oil seeds, and contains around about 96% unsaturated fatty acids and less than 2% russic acid contents as compared to other brassicas. That gives it a pleasant flavor, provides 13% of the total oil supply of the world, and considerable genetic variability for salt tolerance is available. And however, exact sites of salt-induced cellular metabolic damages are not very well familiar to the scientists, and the studies are carried out to explore those sites. Next. Based upon this, we have hypothesized that salt tolerance is a composite trait comprising physiological processes, each of which is controlled by several genes. It is hypothesized that canola germ plasms contain novel sources of salt tolerance, specific physiological mechanism comprising several physiological biochemical determinants. Such determinants might have revealed by several groups of genes, and our main objective was to explore differential expression of genetic elements responsible for salt tolerance in salt-tolerant cultivar Dunkeld of canola crop grown under 200 millimolar NaCl stress. Next. For the purpose, we have gone through this methodology. We grew Dunkeld variety on 200 millimolar sodium chloride stress with other nutrients available to plants, and 2-week-old plants were further used for analysis. Leaves were harvested and quickly immersed in liquid nitrogen and moved to minus 80 degrees centigrade. As an alternative, some leaves were put in RNA later solution to avoid RNA damage. Total RNA from the leaves were isolated, cDNA was developed, and then Next generation sequencing was carried out at Macros in Korea. Then whatever we received were the raw sequences. They were submitted in NCBI. Data quality was assessed using fast QC tool. Adapter sequences were removed. Next. Alignment was carried out using the reference genome. The link is given through BWA software, and the parameters were mean minus T, port minus K, 32 minus M. Feature count metrics, transcript counts were calculated using feature count software using minus N parameter, and biomimetic analysis of feature count metrics was carried out further. Differential expression of genes were determined using bioconductors HR package and R software. DGs were converted to David IDs online using gene ID conversion tool, selecting official gene symbol as identifier and Brassica napus as selected species. Selective DGs with fold change greater than 5 and less than minus 5 were selected for further analysis because we considered that they were more significant. Next. Further, we went for functional enrichment analysis. Only protein coding DGs were selected for further analysis, and selected DGs were subjected to functional enrichment analysis of significant Go terms using G profiler, and FDR threshold was set to 0.05. Then KEGG pathway analysis was carried out, and for annotation was carried out by Koala annotation tool, and get genes using BLAST Koala online, and amino acid sequence data for assigned KO identifiers or K numbers corresponding to individual genes in the genome were worked out. Next. All the data we obtained after NGS sequencing was validated through a real-time PCR, and 5 different genes were selected based on their specific documented role in salt tolerance mechanism, and these were the primer sequences and their gene IDs. Next. These are the details of the RNA-Seq data that was submitted to NCBI, and these raw reads were submitted against these accession numbers. Next. This box plot is actually the raw expression for control and saline plant samples of Brassica napus cultivar Duncan. Next. Basic statistics of RNA-seq data that were obtained for these plants. They included that control and saline had total reads. The total sequences that were obtained, they are given in the table, and total map reads and other details are present in this table. Next. It is a lengthy list. We have selected some differentially expressed genes. I will not go into the details because it will consume a lot of time. I will just read out the main classes, and I request to scroll down all the slides along with it. Some included vascular ATPases, Rubisco units. Next. Antioxidant enzymes. Next. They are some ion transporters. Next. Most of them are upregulated. Next. They are cell signaling molecules. Then glutathione. Next. Glutathione S translases. Next. Next. The certain heat shock proteins were also observed to be upregulated, and different chaperone proteins. Next. There are different heat shock transcription factors that were upregulated, observed to be upregulated in our study. Next. These are different NAC domain containing proteins. They were also upregulated. Next. These were also a good class of transcription factors, MYB transcription factors and their activators. They were also upregulated. Next. These were LIA proteins or late embryogenesis abundant proteins. Then dehydration responsive element binding proteins. They were upregulated. Next. These are aquaporin, different classes of intrinsic proteins that act as aquaporin. They were also upregulated. Next. These are different proteins known as expansins. They are also regulate the water potential of the plants. They were upregulated under stress. hormones and growth regulation factors, and other ethylene responsive transcription factors. They were upregulated. Different serenity, stress related and photosynthesis related proteins and their subunits. They were Some of them were upregulated, the rest were downregulated. These are transcription factors, proline dehydrogenase, PCD or programmed cell death, and next one cyclopropane 1 carboxylate oxidase. They were downregulated. Glyceraldehyde 3 phosphate dehydrogenases were downregulated. Next, all the slides we have gone through are summarized in this figure. If we what we wanted to know was the mechanism of salt tolerance. I have indicated these red colored these genes with red colors. They were upregulated, and those that are shown in green color, they were downregulated. And I have grouped them for the ease of understanding of the participants. That NAC, ERF, MYB, BZIP, ACC, HSP, they were the different classes of transcription factor or regulatory proteins that are meant for the regulation of gene expression. They cause altered expression of other related genes under ciliary stress. Then these TIP, PIP, NIP, EXP, WST, these were different proteins that have to do with the uptake of water and characteristics of cell wall and cell membrane, and they are meant for the osmoregulation or maintenance of the cell wall integrity. Then these are different class of transporters I have mentioned in previous slides. They included sodium hydrogen potassium exchangers and HKT1 potassium transporter hai. Then different calmodulins हैं, B ATPases हैं. They are all meant for the ion homeostasis or making the balance of ions in and out of the cell, or also sodium sequestration. Then the HSPs, LEA, MAPK, PRO, DH, and DI. These are different proteins. They are well documented that they have some specific roles in stress response. And the other category I have isolated were different type of superoxide dismutases, SODs and PODs and CATs. They are meant for the detoxification of different reactive oxygen species or the ions or radicals, reactive species that cause associated when the plant is under any kind of stress other than even salinity stress. The gene ontology, when it was carried out, we were able to classify our information of different genes into 3 different categories. These were including in my molecular process, biological function, and the cellular component. They are presented as a bubble plot. These are the details of some upregulated characteristic Go terms with molecular function, biological process, and cellular component. Important when included antioxidant activity, DNA binding, peroxidase activity, regulation of cellular process, regulation of biological process, homeostasis, iron peroxide metallic process, cellular detoxification, regulation of gene expression, response to oxidative stress, transmembrane transport, potassium ion transmembrane transport, intracellular membrane bound organelles, cellular anatomical entities, and cellular components. Similarly, next, downregulated also included MAP kinase activities, proline dehydrogenase activities. They were in molecular function, in biological function. MAPK cascade was observed, intracellular signaling transduction GOTAM was observed, proline metabolic process was observed to be downregulated. Then cellular component, photosynthetic membrane was downregulated, membrane protein complex was downregulated. These were the K numbers assigned to different protein IDs of the corresponding sequences related to our transcriptome data. After all the verification and outcome of the analysis, it was necessary to validate our data and our findings with the real-time PCR. And these, the primers I have previously shown in my slide, they helped out us to reveal that whatever is in our NGS-Seq data or RNA-Seq data is supplemented by the real-time PCR, and the trend in the both is same. I will conclude my presentation with this slide, that a layman who does not understand any physiology behind salt tolerance, one must understand that the proteins, they are shown in green color. They are moving the ions from outside to the mesophyll cell or away from that. They are different ion transporters. These ATPases, they not only regulate proton concentration, a gradient or pH across the membranes, also they are meant for transportation of different ions like calcium as well. Then they are working in a coordinated manner, and if they are moving sodium out of the cytoplasm into the compartment of vacuole, they are making it less harmful for the cell. That is what I refer to as sodium sequestration, because if it remains in the cytoplasm, it activates other reactive oxygen species and triggers other nutritional imbalance. But when it is dumped into the vacuole, it becomes harmless. And this NXH or sodium hydrogen transporter is the main enzyme which is responsible for, and it was upregulated in our findings, that it is actually dumping the toxic concentration of to the vacuole. And the other important thing is that it is excluding by HKT in the xylem vessel, that it mesophyll cell, which is the main photosynthetic cell, is being kept safe due to the toxicity of sodium ions, reactive sodium ions rather, and they are being dumped into the main xylem so that they do not remain in the mesophyll cell. And all these osmolytes, hormones, stress-induced proteins, heat shock proteins, VIA proteins, they have their definite roles, which are which help them to maintain osmotic balance and different balance in the activities of different enzymes in the cytoplasm. And in the nucleus, these transcription factors are regulating the synthesis of different types of proteins that are required to perform different vital component activities in the cell under this saline environment. And this signal transduction is triggered by the Mendelian and the associated proteins, and reactive oxygen species like SODs mentioned in this are for this quenching of reactive oxygen species. This slide I have made to conclude my findings, that after NGS sequencing and RNA-Seq analysis, we were able to understand all this mechanism. Till this study is not complete, we are working on another salt sensitive cultivar, and we will be comparing the two, and then we will be finalizing our whole mechanism which we wanted or was in our objective. And still we have to learn so many things. Thank you so much. I will appreciate it if you have any questions.